podcast titled Genes, Proteins and Pollutions, whereby we will be exploring the omics in environmental science. I'm Pauline, your host today, and I'm delighted to guide you to a rich and insightful discussion that merges molecular biology with environmental conservation. So our topic today dwells into the powerful application of functional genomics and proteomics, the two advanced scientific disciplines that have significantly transformed how we monitor, access, and manage ecosystems in distress. This molecular level approach have emerged as essential tools for addressing increasingly complex environmental challenges. As many of you are aware, our planet is facing at the converge of pressing ecological issues. The detection of microplastic and polyfluoroalkyl substance (PFAS) and antimicrobial resistant genes (ARGS) in terrestrial and aquatic ecosystems, and even within human biological samples, indicates a level of pollution that often escape the radars of traditional monitoring methods. At the same time, accelerating biodiversity loss, particularly in critical ecosystems such as coral reef and tropical rainforests, raises urgent concern about ecological collapse. And add to that the shifting patterns of zoonotic and vector-borne diseases threatened by climate change, and we are confronted with a multidimensional crisis affecting both environmental and public health. So, in response to this complexity, researchers are turning to omics-based pathology. Functional genomics provides insights into genes expression and regulation in organisms exposed to stressor, essentially showing us which genes light up in times of environmental disturbance. Proteomics, on the other hand, tells us which proteins are being produced, offering a real-time snapshot of physiological and biological stage. So our discussion today will be divided into three main themes. The first one, the genomics is used by studying microbial community and ecosystem. The second one is how genomics aids in pollution degradation and biodiversity monitoring. And lastly, how proteomics can be used to detect early warning signs of environmental stress and the future trends in this field. Joining me, three knowledgeable colleagues, Alia Maisara, Rebecca Sherry, and Priscilla Michelle, who will share their insights and experience. Together, we walk you through the science, the application, and the future of omics in environmental management. So before we dive in, I encourage our listeners to consider how we interconnect our health with the environment. Every breath we take, every drop of water we drink, and every meal we eat is connected to the ecosystem. The more we understand those connections at a molecular level, the better equipped we are to protect to them. So, I'll pass this to you, Alea.
Thank you so much, Pauline. So let's begin to talk about metagenomics. It's a branch of genomics that has truly revolutionized the way we study microbial ecosystems. Traditionally, studying microbes require culturing them in a lab, but it's now known that over 99% of microbial species cannot be cultured using standard methods. So metagenomics overcome this limitation by enabling the sequencing of DNA directly from environmental samples, be it seawater, soil, or air.
Precisely, Alia. This culture-independent approach allows scientists to study entire microbial communities in situ, which rather than looking at individual organisms in isolation. We also can now understand the collective genetic makeup of a community and determine what roles these microbes play within their ecosystem. However, this is also allowed for new insights into microbiome biodiversity, metabolic pathway, and ecological resilience. Over to Rebecca.
To continue, the importance of these microbial communities cannot be overstated. Microorganisms are integral to a wide range of essential processes, including nutrient cycling, carbon sequestration, and the detoxification of harmful substances. These macroscopic organisms act as the first responder to environmental disturbance and serve as sensitive indicators of the ecological change. When environmental conditions are altered, whether through pollution, habitat degradation, or climate-induced stress, microbial communities often exhibit rapid and measurable shifts in their composition and activity. Such changes can disrupt the key biochemical pathways and signal broader ecological imbalances.
That's a great point, Rebecca. Catching those early changes in microbial community really gives a big head start when it comes to managing environmental problems before they get out of hand. We're moving to the segment 2, how genomics aids in pollution, degradation, and biodiversity monitoring. So this one I'll pass to Alea.
So one key area where metagenomics is incredibly useful is in identifying pollutant degrading microbes. For example, in contaminated environments, we often see an increase in bacteria that carry genes for breaking down hydrocarbons or heavy metals. So by using this high throughput sequencing and tools like 16S rRNA gene analysis, researchers can determine not only which microbes are present but also what genetic functions they possess.
However, this is also critical in bioremediation. For example, Pseudomonas rutida has become a model organism for environmental cleanup. Through genome sequencing, scientists also have identified its remarkable ability to degrade polycyclic aromatic hydrocarbons such as naphthalene and pyrene. However, this degradation pathway are encoded also by specific gene cluster that can be mapped and even enhanced through genetic engineering.
Absolutely. And let's bring us to another exciting frontier, synthetic biology.
Yes, synthetic biology and tools like CRISPR allows us to modify or insert gene, the one that we want, into the microbial genomes, giving us custom designed bacteria, which increase capabilities for pollutant degradation. This is an active area of research that blends both genomics with applied environmental engineering.
And I want to add more. So we can overlook horizontal gene transfer. These natural mechanisms enable microbes to share genes related to degradation, resistance, and also stress response. So in polluted environments, horizontal gene transfer often accelerates microbial adaptation, creating ecosystems that are more resilient to chemical stressors.
To add on that, can any one of you give me one of a few example of case study? Anyone?
I can tell the answers. So one of the case study that we look into is the Deepwater Horizon oil spill, which is one of the landmark cases where omic-based tools were applied. Metagenomic analysis revealed a bloom of oil-degrading bacteria in the affected areas. However, researchers identified genes that link to alkane monooxygenase and dioxygenase activity. Meanwhile, the key enzymes in hydrocarbon metabolism. However, the complementary proteomic data confirmed that these enzymes were not just encoded, but however it is actively expressed.
To add on that case, do you know that the sand at where the horizon oil spill happened is called as the Pensacola Beach? These beaches are known as the real world laboratory because after the spill, bacteria like carnivores and marinobacter showed a rapid increase in growth. So the genomic analysis revealed their pollutant degradation potential, while proteomic revealed these studies then confirmed potential functional protein expression, illustrating the combined power of both genomics and proteomics.
For beer pollution, genomics plays a crucial role in biodiversity monitoring. Techniques such as environmental DNA sampling allow researchers to detect species without the need to physically observe or capture them. DNA fragments shed into the environment from skin cells, scales, feathers, or waste are extracted from water or soil and sequenced to identify species composition.
That kind of surveillance can be revolutionary, especially in hard-to-reach ecosystems like mangrove swamp, alpine lakes, and deep sea veins. It gives scientists visibility into hidden biological activities. Anyone have ideas for this?
Pauline, I have idea for this. where the applications are also diverse, such as ranging from tracking of endangered fish species population to monitor the spread of invasive plant species, because it is known as a non-invasive and scalable. However, eDNA also become a preferred method in conservation biology.
Allow me to give the example for that. So one of the case where eDNA has been used is in tropical rainforest, where eDNA has been used to detect invasive species like the Malayan tapir and the Sumatran tiger. It supports conservation efforts by confirming presence without invasive tracking. Similarly, in marine conservation zones, eDNA has detected the return of coral-associated fish following reef restoration. By analyzing specific genetic markers, we can even distinguish between closely related species.
Okay, so we move to the last segment, which is segment 3, how proteomics can be used to detect early warning signs in environmental stress and future trends in this field. So now let's dive into proteomics, which complements genomics by examining what is actually happens in cells at the protein level. So proteins are functional workhorses of biology responsible in catalysis, signaling, structure, and transport. Proteomics also allow us to detect the subtle physiological changes in organisms, and they can expose to environmental stressor. So is there any equipment that we can detect proteomics? Can you guys suggest any of it?
So for like using technologies like LC-MS for MS and 2D electrophoresis, scientists are now able to separate, identify, and quantify thousands of proteins within a single sample. This level of detail allows us to pinpoint biomarkers specific protein whose presence or altered expression can signal exposure to environmental stressors like pollutants, UV radiation, or extreme temperatures. It's a powerful way to understand how organisms are responding at the molecular level, often before any visible symptoms appear.
Besides LCMS or MS and 2D electrophoresis, other examples is known as some well-known biomarkers, which include heat shock protein (HSP), which known as a signal cellular stress, and involves cytochrome 450 enzymes, known as a key role in detoxifying harmful compounds, and metallomanines, which bind to heavy metals. And shift in the level of this protein also can act as an early warning signs in ecosystem imbalance, which often before more visible effects that emerge. So Rebecca, do you have any other example?
Yes, for the things that what Priscilla has mentioned, I do have example for that analysis and the process. A clear example of this is the analysis of the whale blubber, where proteomic studies have detected biomarkers linked to oxidative stress. So similarly, in marine invertebrates, changes in protein expression have been observed in response to stressors like ocean acidification and microplastic pollution, highlighting cellular level impacts well before any changes are seen in entire populations.
I want to add on, as these technologies evolve, we are witnessing the rise of multiomics, an integrated approach combining genomics, proteomics, transcriptomics, and metabolomics. This holistic view enables a much more nuanced understanding of how organisms respond to the environment.
We'll move to another segment, and we haven't ended yet actually. This segment is more exciting development and more forward-looking insights. It's more about emerging topics and innovations. So for this, I will start from Priscilla first.
Thank you, Pauline. And thanks to our advance in machine learning, we are now able to analyze these complex proteomic datasets much more efficiently and with a greater accuracy. So the researcher have developed the predictive models that can actually forecast ecological changes based on molecular signatures before they become more visible in the environment. Am I right, Rebecca?
Absolutely, Priscilla. I do agree completely with her. And it's also also worth mentioning the rise of field deployable biosensors. These are portable tools that use molecular markers like specific DNA or protein sequence to detect pollutants in real time. They are making it possible to monitor environmental health on site, which is a game changer for us and for all the researchers because it allows the researchers to make a quick decision in both pollution management and resource management. What do you think, Alia?
I think it's a very fascinating idea of you, Rebecca. Thank you. So for the adoption of single cell and spatial omics is pushing the boundaries even further. These tools allow researchers to investigate how individual cells function within the context of a tissue, adding spatial resolution to molecular data and enhancing our understanding of microscale interactions.
I really like the ideas from the three of you. So before we wrap up, let's briefly touch on a few of more emerging innovations. So what's the new direction should our listener watch in the omics space? We start from Alia.
So I'd say meta-transcriptomics. Have any of you heard about it? So meta-transcriptomics is gaining momentum, unlike DNA-based genomics, which tell us potential. Transcriptomics reveal what genes are being expressed right now, a more dynamic view. It's like reading the cell's real-time messages.
That's a great innovation. So how about you, Priscilla?
Okay, in my opinion, despite meta-transcriptomics that Alia have said, another promising area is epigenetics in environmental science, which is looks at how environmental factors affect the gene regulation without altering the DNA sequence itself. And the stress-induced changes in the DNA regulation, the patterns can be used as biomarkers of a long-term ecological disturbance. So, Rebecca?
So guys, let's not forget about the rise of AI omics platforms. As these datasets continue to grow in size and complexity, machine learning is becoming essential. These models can pick up on subtle shifts in gene or protein expressions, patterns that could be nearly impossible for humans to do without AI help. That's all from me.
That's a powerful note to add on, Rebecca. AI and biology combining forces to detect early warnings and find sustainable solutions. We move on to the last segment, which is a Q&A session. So I would like to have a quick Q&A session. There's a few of the most common questions we receive from students, environmental research, and curious listeners. So are you guys ready?
Yes.
Okay, so the first question is, how long does it usually take to go on from collecting environmental samples to having a meaningful omics data? Has anyone can answer this question?
I go first. That's a great question. So with today's tools, a basic metagenomics analysis can be completed in about a week, depending on the lab's workflow and how deep the sequencing goes. If you are doing more detailed work, like functional annotations or pathway reconstruction, you might need an extra 1 to 2 weeks.
Okay, and I'm gonna answer the questions, right? And for the proteomics, the timeline can be a little bit longer. The sample prep is more intricate, and the data can be more noisier, which is especially with the environmental samples. You also can typically looking at least 2 or 3 weeks before getting meaningful insights from the data.
And just to add for the both answers of Alia and Priscilla just now, turnaround time is also improving with automation and AI-driven analysis pipelines. Some high-throughput labs are now compressing those timelines even further, especially for standard workflows. But of course, data quality and proper validation still take time, right?
Yes, I truly agree. So, thank you for answering the first questions. So we move on to the second questions. Is omic-based monitoring affordable enough for widespread use, or is it still limited to well-funded institutions? So, would anyone of you like to start first? Rebecca?
Yes, I can answer to that question, Pauline. So it is definitely becoming more accessible. The cost of sequencing has dropped significantly in the past decade. These days, universities, government labs, and even some NGOs are routinely incorporating omics into their environmental monitoring programs. So what about you, Alia? Do you have any idea on this question?
Exactly, Rebecca. So, with this rise of shared core facilities and cloud-based analysis tools, you don't need to own expensive equipment. As long as you have access and some basic training, the barrier to entry is much more lower than it used to be.
That said, affordability still depends a lot on the scale of the project in the region. While costs are coming down, there are still challenges in under resource settings like limited internet bandwidth for clouds, computing, or difficult accessing reagents. So the progress is happening, but it is equitable access, which is still something the field needs to keep working on.
I really like the answer from the three of you. So we move on to the third questions. So can omics be applied in citizen science project? Alea, would you like to start first?
Absolutely, Pauline. So while the actual data analysis still requires technical expertise, citizens can easily get involved by collecting soil, water, or samples using standardized kits. Those samples are then sent to labs for sequencing and analysis.
Good answer. Rebecca, would you like to continue?
To continue on what Alia just mentioned just now, one of the real world project is called eDNA Atlas, which involves volunteers from across different countries collecting environmental samples. The resulting omics data are then used to map biodiversity on a continental scale. It's a powerful example of how citizen science can fit into large-scale ecological research. You want to add on anything, Sila?
Yes, I'm gonna continue from what Rebecca answered. And what's great is that kind of participant also raises public awareness about local ecosystem. When people contribute to real science, they are more likely to engage with conservation efforts, and it bridges the gap between advanced molecular tools and the community-based action.
Thank you from the three of you. So we are moving on to the last questions. So question number 4. What is the biggest myth or misconceptions about using genomics and proteomics in environmental science? Has anyone of you would like to start first?
So for me, in my opinion, one common myth is that omics is too advanced or only lab settings, not practical for real-world applications. But that's just not true. Omics is already being used in fisheries management, agriculture, pollution tracking, you name it.
Good idea. Priscilla?
Okay, for the another big misconception is that omics replace traditional ecological methods. It doesn't. It is actually work best when combined with them. However, we still rely on field observations, species counts, and ecological modeling. Omics just adds another layer of insight, especially at the molecular level. So, Rebecca, do you want to add?
Yes, I would add to that. Some people assume it's all about human and medicine, but not all guys. Environmental omics is a thing. It's thriving right now. So it's helping us understand microbial communities, monitor ecosystem health, and even track invasive species. It's not just a lab tool. It is a lens that do hidden workings of the natural world.
Wow, those are great answers. Thank you all. I think that's really clarify how some common doubts that highlights its versatility and future readiness of omics. So thank you all for such a dual and thoughts-provoking discussions. I genuinely enjoyed the back and forth on the incredible depths of insight each of you brought to the table. What we see today is that functional genomics and proteomics are far more than research tools. They also are foundational pillars in the toolkits of modern environmental science. So, through metagenomics and eDNA, we can now peer into microbial community and access biodiversity with unprecedented clarity. Through functional genomics, we gain insights into stress response pathway and pollutant resistance. And And with proteomics, we move from potential to proof, observing directly how organisms respond under duress. As challenges like pollution, climate changes, and biodiversity loss become more urgent, the integration of omics into environmental policy, conservation planning, and bioremediation will become not just beneficial but necessary. So before we close, I encourage educators, students, policymakers, and scientists alike to embrace omics as a vital frontier in environmental stewardship. The more we decode life at the molecular level, the better we protect the biosphere we all depend on. So we thank you for listening, for joining us today. We hope this podcast deepens your understanding and appreciation of how molecular biology can help restore balance to our ecosystem. Until next time, stay informed, stay inspired, and keep exploring the intersection of science, technology, and sustainability. Until then, bye!
Bye!